Hello everyone, welcome back to my channel. And in today's video we are going to discuss about cell cycle. So cell cycle consists of mitosis and meiosis. So first let us discuss about the mitosis. So what is cell cycle means? The sequence of events by which cell duplicates its genome, synthesizes the other constituents of cell, and finally divides into two daughter cells. So it simply means a division of parental cell into two daughter cells. So cell cycle contains duplication of genome, means if the parental cell want to divide into two daughter cells, it first duplicates its genome, means duplication means doubling the amount of its genome, and synthesis the other constituents of the cell. So after duplicating its genome, it synthesizes the other constituents of the cell, which is nothing but cell organelles such as mitochondria, Golgi apparatus, etc., and finally divides into two daughter cells. So these sequences together constitute the cell cycle. So the cell cycle is itself divided into two phases. The first one is interphase and the second one is M phase or a mitosis phase. So interphase is further divided into four phases. They are G1 phase, S phase, G2 phase and G0 phase. So Z1 phase means it is the interval between the mitosis and the DNA replication. So as we can see in this cycle, so DNA replication starts in the S phase, and this one is the mitosis phase. So G1 phase is interval between mitosis and the starting of DNA replication. So during this phase the cells are active and grows but do not replicate its DNA. So G1 phase marks the cell growth. So during the G1 phase the cell is actively metabolically active and it grows but do not replicate its DNA. So these are the events in G1 phase. And the next one is S phase. So S phase is also called as a synthesis phase. During the S phase DNA synthesis and replication occurs, means the parental cell doubles its amount of DNA, but it increases the amount of DNA but do not increase the number of DNA. So for example, if in the starting of S phase, if the cell has a 2C number of genome, after the S phase it contains 4C, means that total amount of DNA is doubled, and if starting the genome contains only one set, and after the S phase the genome only contains one set, not two sets. So this is the difference between doubling and increasing the number. So here it increases the amount of DNA but do not increase the number of the DNA. So during the S phase DNA replication and synthesis occurs, whereas in animals centriole duplication also occurs during the S phase or synthesis phase. And the next one is a G2 phase. So G2 phase, during G2 phase proteins are synthesized. Means what is the use of proteins? Means the proteins which are helpful for the division of cells, that proteins were synthesized during the G2 phase. And the Z0 phase is called inactive phase or a quiescent stage. Means during this stage the cells do not undergo any division and the cells remain in the inactive stage. See, if we take human heart cells, they do not divide regularly, and the heart do not increase regularly in size. So the heart cells are remained in a Z0 phase. So whenever there is a damage to heart, or whenever we need to replace the heart cells, these cells undergo mitosis. Until then the cells remain in the Z0 phase. So this is about the interphase. So next is a M phase or a mitosis phase. So M phase starts with the karyokinesis and ends with the cytokinesis. So this is an M phase. It starts with the karyokinesis and ends with the cytokinesis. So karyokinesis means division of nuclei and cytokinesis means division of cytoplasm. So by the end of mitosis phase, karyokinesis and cytokinesis are completed and the cells are divided into two daughter cells. So this karyokinesis is again divided into four phases. The four phases are Prophase, Metaphase, Anaphase and Telophase. And the point to remember is mitosis is also known as equational division. Means, so if one cell contains n set of chromosomes. So if this is a parental cell, and these two are the daughter cells, these also contain single set of chromosomes. So this chromosomal material is equally distributed, and the offsprings are same or similar to the parental genome. So hence mitosis is called as equational division. So in mitosis, the offsprings looks same like that of parental offsprings, and the genome is equally distributed between the offsprings. So mitosis is also called as equational division. So let us first discuss about the first phase of mitosis, which is a prophase. So what are the changes that occur during prophase means? So prophase is called as the first stage of mitosis. It is the during this stage initiation of a condensation of chromosomal material, means in the interphase the genome is not present in the shape of chromosome. So the genome is present like a network. So during the prophase, the genome is condensed and they look in the form of chromosome. So this is called condensation of chromosome. So the network like genetic material is condensed in the form of a chromosome. So this is called as condensation. So initiation of condensation of chromosomal material occurs during the prophase, and the centrioles moves to opposite ends, and here the nucleolus nuclear envelope and disappear. So if we take this as a cell, so inside the cell we contains a nucleus. Nucleus contains genetic material. This is called a nuclear membrane. So nuclear membrane and nucleolus disappear during the prophase, and each chromosome contains two chromatids held at centromere. So see, this one is this one is one chromatid, and this one is one chromatid. So this is a structure of chromosome. So each arm of the chromosome is called as a chromatid, and the chromosomes are held together at the centromere. So this central portion is called centromere, and these are the chromatids. So each chromosome contains two chromatids. And the next stage is a metaphase. So by the condensation of chromosome, the prophase completes, and during the metaphase a complete disintegration of nuclear envelope occurs. So nuclear envelope completely disintegrates, and the chromosomes are clearly seen with kinetochore. So kinetochore means, so as I said before, this is a chromosome, and the chromosomes are held together at the centromere. So this centromere contains round disc-shaped structures. Those disc-shaped structures are called kinetochores. So these kinetochores are useful for the attachment of spindle fibers. And the next one is chromosomes lie at equator by spindle fibers, means see, this is a chromosome. So during the metaphase, the spindle fibers from the opposite ends attach near the kinetochore. So kinetochore is useful for the attachment of spindle apparatus. So the kinetochores, so the spindle fibers comes from the centrioles and attaches to the kinetochore. So all the chromosomes lie in a same plane. So equatorial plane means all the chromosomes lie in an equatorial plane. So this is the events that occur in metaphase. And the next one is anaphase. So during the anaphase, the centromere split and chromatids separate. So as I said before, this is a chromosome and this is the centromere. So if we split this centromere, the two chromatids get separated. So during anaphase, centromeres split and the chromatids separate, and the chromatids move to opposite poles. So during the anaphase, the chromatids move to opposite poles. And the next phase is a telophase. So chromosomes decondense and form like mass. So the chromosomes, so these chromosomes again decondense and again forms into network like structures, and the nuclear envelope assembles. Endoplasmic reticulum, Golgi apparatus, nuclei again reappear in the cell. So this is the end of telophase and the end of mitosis. And the last one is a cytokinesis. So cytokinesis means, so up to here we only discussed about the separation or dividing of a genetic material or chromosome. So cytokinesis means division of cytoplasm is called cytokinesis. So if we take in animals, furrow appears between the center. So if we take this is a cell, so in animals the furrow appears in the center and it extends outwards and divides the cells into two equal masses. So, if cytokinesis do not occur, the cells contain multinucleate only in the same cell. So, this is called as a syncytium, which is a syncytium, which is a multinucleate condition. So, the significance of mitosis is it is only restricted to diploid cells, and it is also useful for cell repair. Means if any cells are damaged, so this mitosis is useful for replacing the lost cells. It is called as a cell repair. So it is useful for it is present in epidermis, lining of gut and blood cells, and apical and lateral meristems, and these are useful for the continuous growth of plants. So this is about the mitosis, and this is the diagram. See, this one is a prophase, this is an early prophase and a late prophase. So here the chromosomes are present like network. Here they are organized in the form of chromosomes, and in the metaphase they are all arranged in a single plane, and these are called the spindle apparatus. So these spindle apparatus made these chromosomes align in equatorial plane, and during this anaphase the sister chromatids are separate from the chromosome, and during the late anaphase they move to opposite poles, and in telophase they again decondense and form like a mass or network structure, and after cytokinesis these two cells get separated and two daughter cells and finally two daughter cells are formed from the single cell. As we can see, the daughter cells look like same of that of a parental cell, hence it is called as a equational division or equational division. So this is about the mitosis. So if you have any doubts, do comment in the comment box and do subscribe my channel. Thanks for watching.